Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. So today in this video lecture we are going to talk about another culture media, that is the thioglycollate broth. So in the previous videos lecture we already talked almost 35 video lecture that is related to the culture media. Now this is our 36th video lecture that is related to thioglycollate broth. If you missed the previous videos lecture that is related to the culture media, it will be present, or there will be also a link in the description box, then you will check it. Okay, now in this video we are going to see that what is thioglycollate broth. So thioglycollate broth is again the culture media, which is actually the multi-purpose. It is the enriched media, it is a differential media, as well as this type of media is especially important to find out the oxygen requirement needed for the bacteria or for the microorganisms. Next point says that this type of media is especially used in the diagnostic bacteriology. Okay, why? Because this type of media is the enriched media. Next point says that this type of broth mostly provide or support the growth of anaerobes, aerobes, microaerophilic, fastidious microorganism. Anaerobes, those bacteria that did not need any type of oxygen, and due to this way the bacteria can grow without any oxygen. Aerobes are those bacteria that need oxygen, and with the help of oxygen the bacteria can grow. While microaerophilic, those bacteria that need a small amount of the oxygen due to which it can grow. Fastidious are those that need special types of the proteins, special types of the nutrients, special types of the carbohydrates, etc. And due to this way the bacteria can grow. We call it fastidious organisms. Next point says that the composition of this media. So remember, this media, these are the main composition of this media. Means this media contain sodium thioglycolate, we need 0.5 gram, yeast extract we need 5 gram, sodium chloride we need 2.5 gram, peptic digest of casein we need 15 gram, glucose we need 5.5 gram, L-cysteine we need 0.5 gram, we need distilled water 1 liter. Okay, so we have to mix all these ingredients in 1 liter, so then we will get the simply thioglycolate broth media. These are the requirements that we need in this means in this media, or in this we can say in this to prepare this media we need this requirement. We need thioglycolate broth that is available in a powder form, or we can say dry powder form. We need petri dishes, in this case we need a test tube, not petri dishes. Okay, if you want to make thioglycolate agar, so then we need petri dishes. We need autoclave, we need incubator, we need personal protective equipment like goggles, gloves, overall cap, etc. We need glasswares like conical flask, like beaker, like we need graduate cylinder, etc. We need wire loop, spirit lamp and spreader. Now these are the requirement. Let's talk about the principle, that what is the principle, what is the reason, what is the purpose that we are using the ingredient. The first ingredient that we are mostly using in this is the dextrose peptic digest of casein yeast extract. These types of the ingredient can provide necessary growth factor, and these necessary growth factor are important for the bacterial multiplication, or we can say for the bacterial development. Then we have L-lysine. L-cysteine is actually the special type of the ingredient that is needed for the Clostridium growth, and also need for the other strict anaerobic bacteria. Okay, and in this media, even under aerobic condition we need L-cysteine. Next we need sodium thioglycolate. So this thioglycolate is acting as a reducing agent, and it can neutralize the toxic effect of the mercurial preservative and peroxidize formed in the media, thereby promoting anaerobes and making the media suitable for the test of containing heavy metals. Okay, so this is the importance of sodium thioglycolate. Then we have aerobic bacteria. Okay, so remember, aerobic and anaerobic bacteria can be identified by growing them into the test tube. I will show you these that how will you identify aerobic and anaerobic bacteria in the next slide. Okay, so this is the principle that we need these things, and these are the main purposes. Now let's see the preparation of this media. It's very simple. You need just 29 gram of powder in 1 liter of the distilled water. Then you will mix it. After that, you have to boil the media to dissolve the content completely. After that, you have to distribute the media into test tubes or into the bottle, and then what will you do? You have to sterilize. Okay, it's very simple. First of all, you have to sterilize the media. After that, the tubes will be also sterilized. Then you have to pour 10 ml of the media into each test tube. So now once the media is poured, when it is the media comes to room temperature, then you have to inoculate your bacteria using aseptic technique, and you will incubate your bacteria at 37 degrees Celsius. The result will be that there is a lot of bacteria can grow because it is enriched media. So in this case, the most special type of the bacteria are Clostridium sporangens perfringens, Streptococcus anobaris. This is the bacteroides, Fragiles, Thioglycollate, Streptococcus, Staphylococcus aureus, Pseudomonas aeruginosa, Micrococcus luteus, Streptococcus pneumonia, E. coli, Salmonella, Typhorium, and Eboni. So these types of the bacteria can show turbid growth. And then we have fungi also. Fungi can also grow, like we have Candida albicans. It can also grow, and it can showing fluorescent growth. So this is the growth of the bacteria, and this is the result, and these are the bacteria that are showing perfect growth in this media. These are the identification of bacteria. That how will you identify that either it is Staph aureus, Pseudomonas, or E. coli. So Staph aureus will be showing these types of the turbidity, as you can see. Okay, this is the Pseudomonas, and this is the E. coli. The E. coli turbidity is very high as compared to these two. Okay, now you must have to take this means a blank tube having no inoculum. So due to this way, you will compare all the bacteria very easily. Now this is how you can identify the bacteria, either it is aerobic, anaerobic, or facultative. Let me explain. The first picture is showing that this if you see the turbidity at the top side, and let's suppose if you lose the cap. Okay, if you lose the cap and if you found the turbidity at the top, so we better note that at the top there is a high concentration of the oxygen, and if you found the turbidity, it means that these are the obligate aerobes, because at the higher side the oxygen concentration is high, while at the lower end of the media the oxygen concentration is low. If you found the bacterial colony at the bottom of the tube, it means that these are the obligate anaerobes. Okay, now if you see the bacterial colonies a little bit at the lower end, but more at the higher at the upper means at the top side of the broth, so it means this is the facultative anaerobes. If you see the bacterial colony in whole of the media, and you see the whole media is turbid, this is the aerotolerant anaerobes. Okay, this is how you can identify the bacteria by their oxygen requirement and non-oxygen requirements. Then we have the uses of the thioglycollate broth. So this type of media is specially recommended for the cultivation, differentiation of aerobic microaerophilic, aerobic microorganism, and aerobic microorganism as well. This type of media is also important to check the sterility testing, okay, of the certain biological products that are turbid. The next that we use this type of the bacteria to check the sterility testing of the antibiotic, biological and food materials to determine the phenol coefficient and sporicidal effect of disinfectants. Next, this type of the media is also needed that when we see anaerobic infection is suspected, so then the thioglycollate medium is recommended to isolate the strict anaerobes from the blood. This type of the media is also important to find out the growth characteristics of various bacteria based on their oxygen requirement. So these are the simple uses of the bacteria, the media. Then we have the limitations. So the limitation says that it is essential, this type of the media is essential that you should be freshly prepared or boiled and cooled within 4 hours of the use. This is really important. Next point says that the storage at the lower temperature increase the oxygen absorption. Okay, remember. Then next point says that the thioglycollate media should not be reheated. Okay, why? Because when it is reheated, it will toxic the oxygen radical and are formed on reheating. That's why it should be not reheated again. So this is all about the thioglycollate broth. I hope this video is helpful for you. Thank you so much for watching.